Welcome to the next video of the series. Next video of the series is a hallmark of cancer, which includes evasion of cell death, which typically includes the usual cell death that happens. The programmed cell death is called apoptosis. So inhibition of apoptosis is also a hallmark that will contribute to the carcinogenesis of the tumor. So what is apoptosis? Cancer cells are subject to a number of intrinsic stresses like DNA, dysregulated growth, DNA damage, dysregulated growth, hypoxia, and increased amount of misfolded proteins. When normal cells are subjected to such kind of stresses, apoptosis, cellular mechanisms will trigger apoptosis. However, cancer cells design a mechanism and alter the genes which normally trigger the cell to undergo apoptosis, usually seen in CLL on follicular lymphoma. So how does this dysregulated apoptosis happen? It happens due to defects in the mutations in the p53, reduced expression of caspases, increased expression of IAPs, and disrupted balance of BCL2 family of proteins. So BCL2 family proteins have pro-apoptotic proteins and anti-apoptotic proteins. So either overexpression of anti-apoptotic proteins like BCL2, BCLX1, MCL1, or underexpression of pro-apoptotic proteins like BIM, PUMA, NOX, TMF, or BAX ban will cause decreased apoptosis leading to the cancer. And alternately, there may be impaired receptor signaling pathway. So mechanism of apoptosis in cancer cells is usually by two parts, either by loss of TP53 function or overexpression of anti-apoptotic proteins by members of BCL2 family. Loss of P53 prevents the upregulation of PUMA, which is a pro-apoptotic protein. So what happens? PUMA, which we can see is a pro-apoptotic molecules. Loss of P53 will prevent the upregulation of PUMA because PUMA is produced in less manner. This shift the balance towards pro-apoptotic protein, which causes lack of apoptosis and ultimately leads to growth of the cells. And other part is BCL2, overexpression of BCL2, which is an active apoptotic proteins. How does it do? It will inhibit the mitochondrial permeability. What does mitochondria do? The caspases that are present in the mitochondrial intermembrane will come out and they signal towards apoptosis. By overexpression of anti-apoptotic protein, this function is lost, and this leads to lack of apoptosis in cancer cells. BCL2 mutations are usually seen in chronic lymphoid leukemia and also many other cancers like diffuse large B-cell lymphoma. So examples for BCL2 overexpression, follicular lymphoma. Here what happens? There will be a translocation between chromosome 14 and chromosome 18. So this what does it do? The pro-apoptotic protein BCL2 is overexpressed due to shift in the translocation to the IGH locus. This will cause increase in the cancer. But chronic lymphoid leukemia, it acts to a different mechanisms. So by deletion of 13q, the epigenetic modification happens. A loss of mRNA means suppression of BCL2, which will cause increase in the BCL2, causing increase in the apoptosis of the cell. So this is an example of pathogenesis of follicular lymphoma. If you see normal B cell maturations, the previous B cells in bone marrow, resting B cells, there is BCL2, and general cortical center, they will be so the BCL2 levels are tightly regulated, and this is how certain cells after genetical stimulation, if they are not appropriate for the human body, undergo apoptosis. But in follicular lymphoma, BCL2 mutations, BCL2 is upregulated. Due to upregulation of BCL2, the cells which are actually supposed to die also don't die, and this will cause proliferation of cells, B cells, in the follicle, causing a follicular lymphoma. This is another example of how BCL2 and other molecules orchestrate the formation of things. If you look at here, usually P53 and nuclear factor, whenever they are damaged due to cellular stresses, they decrease the activity of BCL2, which is an anti-apoptotic protein. You see here, by decreasing the activity of anti-apoptotic protein, it will indirectly increase the activity of pro-apoptotic proteins like PUMA, which will lead to activation of cytokine C. Activation of cytokine C will lead to a caspase 9 and caspase 8, which will finally lead to a function called apoptosis. So apoptosis is again two pathways: intrinsic pathway where the is initiated inside the mitochondria by increasing the permeability of the mitochondrial outer membrane, and there is extrinsic pathway, which we discuss later. Intrinsic pathway pathway of apoptosis and mechanisms evade the cell death. The most important mechanisms of intrinsic is loss of p53 function, either through mutation or through antagonism of MDM2, and reduced egress of cytochrome C from mitochondria as a result of upregulation of anti-apoptotic pathway. So these are the mechanisms where the intrinsic pathway is hijacked because of loss of p53 function due to antagonism of MDM2 or increase in the pro-apoptic proteins, which increase the permeability. By this one, the apoptosis is inhibited and the cells grow without any proliferation. So what are the factors that determine mitochondrial permeability? For the first factor is integrity of mitochondrial outer membrane is determined by delicate balancing between pro-apoptotic and anti-apoptotic membranes. The pro-apoptotic membranes, as discussed, are BIM, BAX, and PUMA. The anti-apoptotic proteins are BCL2 and BCL6. There is a third set, which is actually a pro-apoptotic protein called BH3 proteins, which include BIM, BAD, BID, and PUMA. They shift the balance between pro-apoptotic and anti-apoptotic family members, and by neutralizing activity of anti-apoptotic proteins like BCL2 and BCL6, they promote apoptosis. The important thing is PUMA, BIM, BAD, and BID inhibit the functions of anti-apoptotic proteins, and hence they promote apoptosis. So what happens? How do they affect the mitochondrial permeability? When the sum of total BH3 proteins expressed overwhelms the anti-apoptotic BCL2, BCLX protein, so when total be anti-apoptotic, the balance shift towards pro-apoptosis. So BAX and BAC are activated and they form pores on the mitochondrial membrane. So these pores of the mitochondrial membrane, what does it do? It will leak. So this allows the cytochrome will be leaked into cytosol. So it binds to APF1, and which leads to activation of caspase 9, which leads to further activation of other executional caspases. So another check of these mechanisms are inhibitor of apoptosis proteins, which block the inaction of caspase 9. Thank you.